Hey, will you like quicker? Yeah.
No, he's.
Yes.
Oh.
Okay. Don't ask me, we have to call you, yeah, and it's all good. Nothing really, that happens.
That's cable.
Alright, we'll give it very excited until he comes back with a clicker.
And you said.
Perfect. Alright, I think we're gonna get started just because we do have that card out. We'll start trickling out at 10 till because there is a class in here at 5 and then we definitely have to be up by 5 till. And then our social is going to be right down the hall in 2. Remember number 30. 30, thank you. Wherever that is. Yeah, so 2:30, so it's right down the hall is the eco-social. So if you do have questions that don't get answered, if Justin's still going this presentation right up to that last minute, or there's enough questions, you can always socialize afterwards and chat about. However, I am not going to introduce him as I don't really know. I met him like 5 minutes ago, but David does know him, so I'm gonna turn over the floor.
Thank you. Hi everyone, welcome to this semester's second eco series. I'm not mistaken, number 2. Yeah. I'm gonna introduce one of my committee members, Dr. Justin Luong. He is an amazing rangeland and just ecosystem scientist, so incredibly knowledgeable and a great mentor to have. And I don't want to butcher his own bio, so I'm going to read off the script. Dr. Justin Luong is an assistant professor of working lands and restoration ecology. He studies rangeland ecosystem responses to climate change, species invasion and conservation management, and interdisciplinary methods that combine field, greenhouse, and social science approaches. His work focuses on biodiversity, endangered species conservation, grazing management, functional traits, restorative agrovoltaics that I just learned about, bridging the science and practice gap, and plant communities and restoration project responses to climate stressors and varying land management practices. So yeah, super knowledgeable on so many things, and just a really great person that just knows how to combine all of these subjects and makes it really easily digestible. And yeah, I can't speak enough of like how much he has helped me in my project. And I know other students, other rangeland students feel the same. And also I'd like to both thank and congratulate Dr. Justin Luong for it was his efforts that helped get our rangeland science program here at Poly Humboldt accredited with the Society of Rangeland. And if I'm not mistaken, we are the first university in California to receive that accreditation, and only one of 15 schools in the entire world to receive that accreditation. So it is a huge honor, and I can't thank you enough. Yeah, thank you all so much for coming here. And I hope to see you all at social afterwards. But with that, I will give it to you.
Thank you, David, for those kind words. My name is Justin Logan. I'm an assistant professor at UC Berkeley at Working Lands and Restoration. And thanks for having me here today. I should have titled this "Why You Should Be a Range Major." Still can change. You could change still. But anyway, I'll be talking about solar microgrids and how they generally have minimal effects on soil properties, but they actually do. I should have changed the title. They do have effects on plants. But yeah. Before we get first.
Click on the screen.
Yeah.
Oh yeah, you have to.
Yeah. Before we get started, I just want to do some brief acknowledgements, and I want to start by thanking our end use partners who supported us by allowing us land access. And so Marianne Bethel from the Redwood Coast Energy Authority, Elijah Moulton from Redwood Empire Public Television. They have a broadcasting station up in Neelan, as well as Marianne Bogdani from Lazy J Global Home Parks. And numerous members of my lab that helped with data collection and processing, especially Sean Dillon, Angelina Garcia, and Max Shea, who worked really hard this last few days to get in all of our data from this last 2025 field season. So I could update it for today. Sean also is an amazing artist who has illustrated a lot for us, as well as a few other people. A lot of this work takes place on Wea and adult tribal territories, which I also want to acknowledge. I also want to mention that a lot of this work was led by student researchers through the Learning and Live Employment Program, Gabriel Abundis, Logan, Holey, and Claudia Alfaro-Hernandez, who all have also received grants to work on this project as well. And of course, we thank all of our generous funding support too. So California rangelands are essentially, rangelands to start with are uncultivated habitats that support wildlife or livestock grazing. And so they're pretty much all habitats that aren't agricultural and aren't densely forested or urban. So like I mentioned, if you guys are already rangeland ecologists, then you should take your majors. Rangelands take up 60% of land area globally and approximately 25% of land area in California. What is happening? All right, I guess you guys can all get a preview. Okay, I'm sorry. I don't know what happens.
How the monitor's turning off?
I can't just, luckily we were on. It must be the clicker stuck.
Just want to use keyboard, that's fun.
Sure, I'll try it over time. It's great.
Yeah.
Yeah, okay. Never happened. It's all right. I guess I need to show screen again too.
Reports.
All right, taking it's not. Okay. Okay.
Thank you, appreciate it.
Where were we? I better not think too much. Okay, so California rangelands support one of the largest global economy, California, which is the fourth largest economy in the world. And agriculture, rangeland related products support the second and 10th top grossing product in our state. Rangelands also provide diverse ecosystem services from flood attenuation, soil carbon stocks, biodiversity and wildlife habitat, and much more. And they also vary widely across regions and within a given habitat. Rangelands are also really important in thinking about climate resilience and supporting nature-based climate solutions, which is a major goal for our state and globally with the United Nations. Rangelands are also really important in California for biodiversity. California is one of 36 biodiversity hotspots in the world, and California rangelands support 40% of our plant species in the state, and that's nearly 2,600 plant species. They also support numerous wildlife species, many of them which are listed, and I think from the last, maybe it's been updated, but when I last read, they support 90% of the state's threatened and listed endangered wildlife species. They're also really important pollinator habitat and refugia, and oftentimes we're finding bumblebees, especially native bumblebees, typically prefer a lot of the native forbs that exist in restored sites, as well as in more of these native remnant sites compared to areas that have been heavily invaded by annual grasses. The plant diversity that rangelands support is really important for ecosystem services. Diverse plants provide more functioning. They also provide what we call redundancy in the ecosystem in case you lose a species. There's another one to take its place and to support that function. Plant diversity also helps support animal health and to minimize production costs by reducing the need for supplemental minerals or feeds. Many studies have shown that diverse forbs have different mineral contents and nutrition, which kind of help the support micronutrient requirements for livestock and wildlife. So there's a growing demand for clean energy though, and California supposedly is on track to achieve 100% clean energy. Solar energy, of course, as we all may know, can help meet these energy demands with lower carbon emissions. And nationally, at least it used to be our goal to convert 5.3 million acres to meet the Department of Energy goals by 2035 to increase the amount of clean energy across our nation. In California specifically, which is the fourth largest energy producer in the country, our goal statewide is to have completely clean energy by 2045, which is only 20 years away. And so thinking about strategies where we can improve or integrate clean energy in a way that doesn't damage our important biodiversity on rangelands or potential for livestock production can be a really important point. And a lot of times this can be a major issue because most of us have probably seen solar panels like this in pictures, or if you've driven out in the desert, where there are these huge solar farms paved over in the desert, which massively affect land area. They urbanize this habitat. You lose a lot of biodiversity, wildlife habitat, and the soils change, and you also have more flash flooding and less groundwater recharge. And these large solar farms definitely have major issues. And so we want to think about how can we think about creative solutions for that. And because of that, these large farms can also cause additional ecological, human, wildlife conflict issues. When we lose a lot of habitat, we lose forage for wildlife and for livestock. And so there's going to be less area, and so wildlife and livestock are going to interact more and cause issues for land management, landowners, as well as likely advocacy groups. Solar microgrids are potentially a unique and creative way that can lower the impact, development impact on a landscape. And you can kind of see these solar microgrids can kind of fit within a landscape and be a little bit more flexible and help support local energy reliability and sovereignty, and also help with energy demand during emergencies when the power grid might be overstrained. And so I think they also, one of the reasons they have some of these solar microgrids out of the airport, for example, is to support energy reliance in case something happens. However, like I mentioned, we don't want to lose the valuable ecosystem functions and services that we get from rangelands. And so we want to think about how can we optimize the ecosystem services with the proper management when we put these solar panels out into the landscape, so that we can still have energy production and also still support livestock production, as well as native plant biodiversity and pollinator habitat and wildlife habitat and more. So we've been talking a bit about the microgrids, and so these microgrids, as you can imagine, as you kind of see in this last picture, kind of have an effect on the environment. And so there are these open areas in the field here. There's the areas right beneath the bottom of the panels where it's most narrow, and then in between the panels right here at the center beams where it's probably most shady, and all the way at the back under the top of these panels. And so you might expect that these different areas might increase the habitat heterogeneity of the environment, so the variability of that ecosystem, and create different types of microhabitats that maybe could support more diverse communities and functions in thinking about it at a patchwork to landscape scale. And so based on some of that shading and some of that other stuff you might expect, you might think then that solar panels or microgrids will likely also influence abiotic factors, so for non-biological conditions, that may then result in changes in biological interactions as well. And then different positions in the microgrid may differently affect environmental conditions. And so in the bottom position here, or let's start in the field position, which is kind of out in the center. And so there you might expect that it's really open. You might have the most sunlight there, highest exposure to wind, and maybe the lowest soil moisture because it's out in the open. You might also expect the lowest soil compaction in that area, and that plants that are adapted to that habitat typically exhibit traits that are more drought tolerant. When you go back one habitat to the bottom, You might expect there might be increased rainfall as rain hits the panel and goes down, which may also result in erosion. And then there may also be a change in light, because we might have increased shade in this area. As we go back a little bit more to the center position, we might expect there could be more the highest levels of shade since it's right in the center of the structure. There may be the lowest amount of rainfall since the cover is right over it, and likely also the lowest wind exposure and light exposure. And so plants there likely need to be adapted to shady conditions that maybe don't receive a lot of direct rainfall or sunlight. In the very back, when you get to the top position, you might expect that there again is a lot of shade from the panels, which might expect where you might expect plants to grow taller to get more light. And there may be reduced rainfall because the water might hit the top of the panel and go down. Plant functional traits are measurable characteristics that relate to plant function and ecosystem services that co-vary with environmental conditions. And so they're a perfect tool to evaluate how plant communities might change in these different microhabitats along a solar panel. Because again, they have established trait to environment relationships. So for example, some plants might have really high, might be really tall in the shade to stretch to get light. Whereas plants in the direct sunlight in the field position might be shorter and might have thicker leaves to minimize water loss. And they don't really need to stretch for light in those cases. And so these traits can generally inform what plants are best suited for these different microhabitats and their potential contribution to ecosystem services. Soil biogeochemistry is also important to understand because we care about soil properties and soil health, how that affects plants, and how that also affects our soil carbon stocks, again, to address important nature-based climate solutions. And one thing you might expect is microgrid development or development in general could impact and cause soil compaction or increase bulk density because you have to take out bulldozers, or I don't exactly know how micro panels are installed, but you still probably have to have something heavy out there to put them in. Different microhabitats can also support different plants based on the different environmental conditions, soil properties like organic matter or soil moisture that it supports. With all that in mind, we had three main overarching research questions. The first was, how do solar microgrids affect rangeland plant communities and functional traits? We're also interested in how ecosystem services are impacted by different microhabitats along a solar microgrid. And how do solar microgrid installations affect soil biogeochemistry? We have 3 study sites across the county, one in McKinleyville at the airport, one in Arcata at the Lazy J mobile park home, which you may have seen driving north on the left side of the 101. And another one up on Neeland at Berry Ridge, where they have a broadcasting station. And so it's a loose elevation gradient, but really the main reason we have more than one site is because when you only have one site, it's really hard to make broader or more general or broader generalizations. And you are restricted to making recommendations or generalizations that just apply potentially to that site. Having more than one site allows us to actually indicate whether or not these trends are can be transferable across the region or into other areas. These are all Humboldt County rangelands, so they all support grazing in some way. Insects also graze, so don't forget about that. Maybe you're interested in insects, another reason to be into rangelands. Coastal prairie habitats are these are all classified as coastal prairie habitats, which are grasslands that typically receive summertime fog, and that summertime fog helps increase shade essentially and reduce heat stress, and reduce evapotranspiration. Some plants can also capture fog drip on their leaves, like redwoods, but also grasses and other very low leaves, and drip that down to their root zone. These areas also all have different management, so we can better understand, well, are there trends that hold true regardless of management or site? In particular, the lower elevation sites at Lazy J and Neeland are managed annually. ACV or the airport is the most heavily managed with several mowings per year. Lazy J is mowed twice per year, and Neelan is not managed like with mowing or any type of external management at all. So first thing we did was, well, not the first thing, but one of the things we did was measure the environmental conditions of the sites. And so we went through, we measured the photosynthetic photon flux density, also known as PPFD, using a LIGO barometer near solar zenith, which is solar noon, which sometimes varies by season. So for example, in the spring, we have a lot more daylight, so solar noon might actually be closer to 1 o'clock. Whereas in the winter, we have a lot less daylight and sometimes solar noon can be closer to 11 a.m. We conducted in situ soil moisture measurements with a Campbell scientific probe to measure volumetric water content. And so you can see us, Logan measuring PPFD down here, Claudia measuring soil moisture up top. And you can actually see that it is completely flooded and where here is very in the water. So these environmental data were taken at snapshots at the beginning of spring and summer 2025 at this change of the season. We plan on taking these measurements as well in the fall and winter to have a full year of data. I'll also mention they were taken in the same design as our plant data, which you're going to see next, but essentially in every single microhabitat several times. We measure plant community composition annually in May, which is when peak growth or biomass occurs, because that's when plants are flowering and you can identify them. And that we did that in 2024 and 2025, and maybe we'll do that also this upcoming field season in 2026. We measured in the 4 microhabitats I've been talking about, in the field position, the bottom, center, and top. And at each of these sites, we laid out 3 transects that intersected with 6 rows of solar panels. And we put down 1/4 meter quadrats at each of those 4 micro positions along those 6 rows of microgrids along the transects, totaling for a total of 18 quadrats per microhabitat at each site. We measured plant traits, leaf dry matter content, specific leaf area, leaf lobeness, and plant height from the most dominant species that make up 90%, 95% of total plant cover. I won't bother you with the boring specifics of those plant traits unless you want to know about it later, but these traits are typically related to drought resistance, grazing palatability, competitive ability, and growth rates. We sampled the most dominant species in 2024, but we did a lot of sampling. We sampled, and I forgot to correct this one, we did population level sampling for 5 individuals, not 7, by microhabitat and by site. So for example, we collected leaves from up to 20 individuals of a single species at a site for as many species that we needed to. And over the last 2 years, we collected leaves for this project, from 1,500 plants. So we next combined the community and trait data. Here's Carl. And the plant community and trait data were again combined with boring matrix math to create community weighted mean trait values. And this is important because it accounts for the abundance and species dominance in the ecosystem and their potential contribution to ecosystem functioning and services. And so, for example, we don't want to say a species that has 1% cover has the same contribution to the traits and their effect on the ecosystem as a plant that has 80% cover, like an invasive species. And so that's why we weight the trait data with plant cover data by species. And then if you care about this, we analyzed most of our plant data at the plot level, so that's by quadrat. And we also summarized it based on origin, so native, non-native, and life form, grass or shrub. And a borb is a non-woody wildflower, if you're not unfamiliar. And maybe some other things. We did some soil sampling as well just in 2024 at 2 depths, at 0 to 5 at the surface level and from 5 to 10 at the shallow level. And again, we collected it in the same positions as we did for the plant community data and for the environmental data. And we used a rapid soil core sampling method that hopefully will work here, which uses a power drill developed by Dr. Carrie Byrne in ESM. And we basically drill down, and this is normally used to make doors. And then we extract all of the soil, and we can get a known volume to calculate bulk density. And again, we collected this from all of the 4 microhabitat positions with 4 samples per depth at each site or along the transects. After we collected those soils, we processed them for soil bulk density, which is a gruesome process, which starts with the soil, and then it goes through a series of sieves, and then we separate out rock fragments, and then roots, and the soil. The soil itself is then used to calculate bulk density. We separated, as I mentioned, coarse fragments or rocks and root production. Roots essentially sit at the top of the sieve, and people sit and they pick them out with little tweezers for 1 to 4 hours until they're done. So it's a lot of work. And then we do something to measure soil moisture and soil carbon using a method that we call loss on ignition. And so we take these soils at known mass quantities. They start out brown and they turn more red as they oxidize and heat up. We weigh out 3.000 grams of soil plus or minus 0.00001 gram of soil. And then we bake it at different temperatures and we measure the mass loss. And that tells us the soil moisture, soil organic matter, soil carbon, and soil inorganic carbon. So just a reminder, here are the 4 positions again, the field position, bottom position, center position, and top position, which nicely match the colors that you're going to see on the figures coming up in the next slides. All of these results were analyzed with fixed effects linear models with a random effect for site. And if relevant, we used also a random effect for season for the environmental data and a random effect for year when we had multiple years of data. These data were log transformed for analyses to meet parametric or normality assumptions, and then back transformed for visualization. Random effect, if you're unfamiliar, essentially tells the model that it started each site or whatever factor you're saying is random has a different starting point. And to analyze the relative change of different traits or factors within that kind of framework, and then average the relative change of these kind of different starting points all together. So that way we can incorporate all these different sites together without having a major concern about how different the sites are and how that might affect our results. All right, so the first thing we did, or I'll be showing you, is how microhabitats influence abiotic site conditions, and this work was led by a range student, Logan Holey. And so the first thing here that we have on the always on the x-axis, you'll see the site, airport, Neyland, Lazy J, and it'll always be in the same colors. Bottom is going to be in blue, center in pink, field in gold, and top in green. On the y-axis is going to be the various traits or measurements that we assess. This first one is photon flux density. And we can see here that PPFD definitely varied by microhabitat. So the amount of sunlight that plants can use varied. And the bottom and field positions up top did not differ from each other, but they were both significantly higher than the 2 center and top positions in the center and further back. And the top position also had more light than the center position. So that very back spot had more light than the spot under the panel. Then we looked at air temperature and we found that there was essentially a trend going backwards from the field bottom, center to top. Air temperature was highest to lowest. So highest in the field, then the bottom, which also received some direct sun exposure. And then it was higher in the center position and then lowest in the top position in the very back, where potentially it was higher than the center position because there's a lot of space and airflow, and so it can potentially get cooler. And then we looked at soil moisture with the soil moisture probes, and we saw that soil volumetric water content did differ, and that the center position, in the middle, had the highest volumetric water content on average. And the top positions also typically had more volumetric water content compared to the field positions. And that makes sense because again, these are the areas that we would expect more shade in. A lot of this soil work was led by ESM student Claudia Alfaro-Hernandez. And we found that soil microgrids don't really strongly influence soil physical properties. We just looked at soil moisture on this last figure, and this was using a probe that cost a pretty penny. And we then also, like I mentioned, did loss on ignition to measure soil moisture. When we did loss on ignition, we actually didn't find an effect of soil moisture or a change in soil moisture with microhabitat or depth. So loss on ignition is known to be a little bit less sensitive. And so we could probably expect the soil moisture probe data is more accurate. And this may also tell us that loss of ignition may not be appropriate to detect soil moisture changes. Another reason could be because we collected these soil samples at the end of the spring, kind of in early summer, when the soils might have already been kind of dry throughout. We looked at bulk density, which again is how compact a soil might be. And we essentially found that bulk density did not really change across microhabitats, which is a good thing, which means that development of these panels likely did not affect compaction. That comes with the caveat that the field position could have already been impacted because it's in the middle of all these panels. And we didn't measure soils outside of the panel region. We also found that soil coarse fragments, or the relative amount of rocks, so to speak, did not differ by microhabitat or depth. We also found that microhabitats influence bulk soil carbon, and reproduction, but not inorganic carbon. So the soil organic carbon, again, we in this case we measured bulk soil carbon, which is how we measure soil carbon stocks and how we often account for soil carbon for carbon credits and carbon farming, etc. And so that's calculated by multiplying the depth of sampling by the percent of soil organic carbon by the bulk density. We essentially found that bulk soil organic carbon varied by microhabitat, where it was lower in the bottom position compared to the center position, and also marginally lower in the bottom compared to the top position. This is where you get likely with the rain coming down and hitting the soil. So there could be a lot more erosion, which is eroding away fertility, as well as soil carbon, which means that could be an area very sensitive for soil carbon loss. Bulk inorganic soil carbon didn't differ by microhabitat or depth. Root production, though, did vary by microhabitat and depth, and the bottom microhabitat had the lowest root production, kind of also consistent with this potential effect of erosion happening in the bottom area. This could also be the other reason why we're seeing less soil organic carbon, is because there's less roots. Center position also had produced marginally less than the field. And this is likely because it's shady and there's not much plant growth in these areas. There was more roots in the shallow zone, 5 to 10 centimeters, compared to the surface level at 0 to 5, which is not necessarily something that we expected. We found that microhabitats influence plant and ground cover types. This first plot is of native plant cover. We found that native cover varied by microhabitat, where the field position had more native cover than the top and center positions, and the bottom positions also had more native cover than the top positions. And this seems to reflect the trends of soil moisture and temperature, where the native plants tend to be occupying areas that are drier and warmer. Because the field position was the hottest spot, it was also one of the drier spots. The bottom position was the second warmest spot, and it was also the second driest spot. Non-native plant cover didn't vary across microhabitats except for the center position, where it was lower compared to all other microhabitats. And that is likely because it reflected total plant cover, and our plant cover was typically dominated by invasive non-native species. And so it's basically showing the same trend as total plant cover with reduced plant growth in the center position. And that's probably because there's more bare ground in the center position compared to anywhere else. And so we're not really having a reduction of invasive species in the center position. We're just having a reduction of total plant growth and an increase in bare ground. Otherwise, bare ground didn't differ by microhabitat. And this work was led by Gabriel Abundis, a range student as well. This one as well. Microhabitats influence plant life form distribution, and so we looked at grass, orb, and woody plants. So this first one is grass. And we found that grass cover was lowest in the center microhabitats, mimicking total plant cover. And most of our non-native species were grasses, so that makes sense. And so we don't really know, this doesn't really seem like a true effect. This is just an effect mimicking total plant cover at increased bare ground. That is already grayed out for some reason, but we also found that rushes and sedges were all native. All the rushes and sedges we observed in our study were native species, and they only occurred in kneeland. And so they essentially showed the same trends of natives as native plants, with the highest cover in the field positions and bottom positions compared to the other ones. Forbs, again, are wildflowers, not grasses and not woody. And they differed by microhabitat, and the bottom position had more forb cover than the top position and center position. The field position also had more forb cover than the top microhabitats. This seems to also be mimicking native cover, where these forbs tend to be occurring in some of these drier spots. And a lot of our forbs that we observed were native. And then finally, we also looked at woody species with the caveat that there were no woody species at Lazy J. But we saw that woody species cover typically increased in the center positions compared to the field. And this is likely because there's more moisture in the center positions and more shade. Woody plants are typically more shade tolerant and typically require more soil moisture. This can be an issue or problematic because a lot of the woody species that we observe were non-native blackberry, and those can become very viney. And if you think about it over time, these vines in the center position could grow up through the gaps in the panel and cover the panels and reduce energy capture. This next part looks at microhabitat influence on plant traits, and this was led by Logan Holey in the range program. And we looked at first plant height. And again, we're looking at community weighted needs. That's what CWMs are. And that again is accounting for the relative cover of these different plants and the plots. We essentially found plant height differed. It was higher in the top compared to the center and field positions. So some of that makes sense. The top position, you would expect the tallest plants because it's very shady back there. You would also expect shorter plants in the field and bottom, because it's very there's a lot of direct sun, so plants don't need to stretch for that light. We also saw slightly shorter plants in the center position, and that could just be because maybe they're just not getting enough light at all to grow taller, and don't have the energy to stretch as much because they don't have enough light there. But I'm not 100% sure. We also looked at leaf lobeness. Leaf lobeness is essentially the potential for a plant to cool itself at the leaf level using surrounding air instead of transpiring water or wetting water, so to speak. And so the more lobed a leaf, the more surface area contact it has with the wind. And so it minimizes kind of transpiration water loss. And so we see here that the top position has the highest leaf lobeness. And lobeness was lower in the center positions compared to the bottom and field positions. This could be because there's more moisture in those areas and less sunlight, so they don't need to cool themselves as much. There's also cooler temperature. In the back areas where the plants were you wouldn't necessarily expect high leaf lobeness where there's a lot of shade and plants are already cool. But maybe one reason we might see higher leaf lobeness is because the plants are growing taller, and because they're growing taller and there's more roots, they might be using more water. And if they're using more water, they need to find a way to reduce that water loss. We also looked at leaf dry matter content, which is essentially a measure of drought resistance. It's a scale from 0 to 1. 1 is the most drought tolerant you can be, whereas 0 is the most drought sensitive you can be. And leaf dry matter content did vary by microhabitat, where we found plants in the center position, where it was most shady and moist, had the lowest leaf dry matter content, indicating the lowest drought tolerance and the highest drought sensitivity, which again makes sense because there's less drought stress there and more moisture available.
active.
So we just went over a lot of stuff. So I have tried to summarize everything for us neatly in this little figure. And it essentially kind of goes like, I think of it as a gradient, although the bottom is down here, it's just to make it look more clean. I think of it going from field, bottom, center to top. So in the field position here, in that open gap, in summary, we find that plants, the field position is typically the driest and hottest microhabitat. And it typically supports shorter plants that tend to be native plants and forbs and sedges and rushes. This is also the area with the highest root production, potentially because of these native plants. We then go back to the bottom position, just a few steps backwards, where you still get direct sunlight in this area, but you start to get some shade. But then you have also the effect of water running down in this area. It's still dry, it's hotter, but not the hottest. The field is the hottest spot. And it has less roots. And because of that, it could cause less soil carbon and potentially more soil erosion. And so that could be a potential place to think about management or seeding efforts or just for further study. Then going back, crawling under the panel, you'll be in the central position. This area is the moistest, has the least amount of sunlight, and has the most woody and cover as well as bare ground cover. It has less invasive plant species and less grass cover, but that is likely associated with total plant cover, a reduction in total plant cover. Plants in this area tend to be shorter, had low leaf lopeness and low LDMT, indicating that they're likely drought sensitive in that habitat. And so we may also have to, for example, if we were going to think about seeding or introducing native plants, you might be able to get away with using more drought sensitive plants in that specific spot where it may not survive in other areas. And then going all the way back to the top, we see that plants there are the tallest and they're the most lobed. They're stretching out to get light, potentially becoming more low to help retain the water as they stretch and have greater amounts of leaf to lose water from. These areas have less sun and tend to have a little bit higher soil moisture and have comparable production as the field area, the field zone. Okay, so overall, this means that solar microgrids, of course, as we might expect, transform environmental conditions, but some of this can't be managed. There's different areas with different amounts of light and different soil moisture. We could feasibly run out drip line, for example, if we're investing in doing some restoration efforts at these areas or trying to introduce high quality forage plants in these areas. If we know they're drought sensitive and we want to have a uniform introduction across the landscape, or we could think about how these different environmental conditions favor different plant traits and then selectively use those plants with those appropriate traits in those specific microhabitats. We may also want to think about, for example, how these environmental conditions affect ecosystem services. Like in the center position, there's less forage produced, and maybe we do want a lot of forage produced there. And so maybe we need to find a way to help plants grow better in that area or to introduce plants that can handle that shade and that low light level. We want to think about how we can protect soil in the bottom area below the solid solar panel to reduce erosion and maximize soil carbon retention. And we also want to think about how we can improve native plant cover in some of these areas that don't have as high of native plant cover, like in the center and top positions, which are typically dominated by more competitive plants, which tend to be non-native plants. Certain plants, microhabitats favor plants with certain traits and certain life forms. And again, it seems to be related to these conditions where native plants like drier, warmer, or maybe they just do better because non-native plants can't do as well in those habitats. And we do see that plants, there's something adaptive happening within the community with their weighted trait values, where plants are exhibiting adaptive traits depending on their position with their plant height, their leaf loadness, and leaf dry matter content. I also mentioned that, because we didn't see much of a change in bulk density, that may also indicate that, these types of developments don't strongly affect, soil compaction, which is hopefully a good sign. But I mentioned that with a caveat that we measured within the field position within the solar microgrid, not outside the solar microgrid. We're still working on this project and there's a lot of really cool things going on. So Elias Sandoval, a range soil student, is working or has actually built from scratch custom air temperature sensors that are programmed with Python, custom Python code, and with all of these crazy little electronic parts and soldering that we have now deployed out at our VLAN site with wires that are taking continuous air temperature measurements. Bailey Glashon, who is a range student as well, is conducting insect surveys or over the summer put out hand traps and pitfall traps out in the same micro position that we've been doing plant community sampling, soil sampling, and trait sampling, so we can better understand if there is a difference in ground dwelling insects in these different microhabitats. And Logan Holey, a range student, will be using these cute little R sensors out also at Neyland to take continuous light measurements, kind of like instead of those snapshots, so we can see if we have a better understanding if we do continuous measurements as well. And so there's a lot of upcoming and ongoing work with this project. And with this, of course, I want to thank our amazing authorship team and Poa, the puppy, Poa's a grass, and a dog. And all of our amazing contributors to this project. And thank you all for coming, and I'm happy to take any questions.
Thank you, Justin. That was awesome. So sorry about the issues there. The first preview of the entire presentation. I think we have like 5 minutes for questions, 4 or 5 minutes for questions. We have a hard at 4:50, but we'll be in WDFS 2:30, just around the corner for more questions.
So back at home, they're installing panels that move with the sun.
Sure.
How, very broadly, how would you expect that to change your microclimates?
Yeah, that is a lot more complicated, and I couldn't say for sure, of course, but I feel like in those cases, there would probably be a lot less effect of a microhabitat, and there would maybe be effect just centered in the center position, where you would probably expect the highest change in environmental conditions. Does that help?
Yeah, I was just curious if you
Okay.
would think that like the edges, since they still get more shade than normal, than the field.
they will, yeah.
Be like a gradient maybe?
Yeah, it could be, and it kind of depends on how they're angled and with like the way the sun is. But generally, from what I understand of those systems, they usually move to capture the sunlight. And so I would expect that instead of having like when we look at that the conceptual figure, it almost runs like a transect. And as you step backwards, the temperature changes and things like that. Whereas in that case you mentioned, it's kind of swinging with the sunlight.
Yeah.
And so it's like one area is going to be continuously shaded. There probably will be edge effects, but unless you do really kind of fine scale sampling, probably wouldn't detect that.
I'm curious, you mentioned a couple times about doing restoration at these sites. Is that restoration for specifically the disturbance of the swallowing soil minerals, or is that restoration that targets in the invasive species trying to bring back native communities to the farmland?
Well, kind of whatever people want to do, because resources kind of vary by land manager, and it's none of these, a lot of those things, I mean, invasive species removal costs the least, seeding is okay, and then planting plugs is very expensive. And so it really depends, and the first step is to really understand what plants will do well, so we don't waste money. And so that's kind of like what this whole process is for. And then the next step is now to then, hopefully, what I'm most interested in is doing seeding or planting. I do invasive species management as well, but it's less fun. So it kind of depends on the land manager, the context, and what. I don't know if that answers your question.
Yeah, I guess I'm curious sort of like their goals might be, it's to make the best rangeland under solar and best rangeland.
Well, I think it depends on the land manager, and some people are really interested in maximizing ecosystem services, supporting pollinator habitat. There's a goal of support pollinator habitat with more native plants. And one other thing to consider is some native plants can attract a lot of birds. A lot of birds can also cause issues because they shit on panels, power wash, or reduce the efficiency of energy capture. And so there's a lot of balancing and thinking that needs to be done before we can actually really effectively introduce native plants or more plants into the landscape in my experience.
No, if it's an issue, there are issues when it comes to heavy metal contamination on these kinds of grids installations.
That's a great question, but I don't know because I don't measure heavy metals. And I haven't read it in literature, but if you would send me an email, maybe I'll have an answer for you one day.
Were there not any samples taken from like outside of the grid, like specifically just the grid habitat on the sites? Like, cause if the rest of it is also range where the grids are situated, why wasn't that data like included? Well, that just, was it just like a limitation?
Yeah, it's just a limitation, that would incorporate a whole nother additional design that we would have to incorporate, and this work, took a lot, it's a lot of labor. And so that's probably the biggest limitation. Some of these areas, we could do that, other areas we also couldn't have a really good equivalent comparison, and so we decided not to, couldn't do it. Like at Lazy J, there is no adjacent parcel that you could do that first. And here at Neeland, it's grazed right on the other side, so it's also really different.
Awesome, thanks so much again, Justin. Well, our eco social over just around the corner, if you wanna follow us there, we'll walk over right now.